Everybody, going to give you a little video lecture here on some of the topics for today and introduce the case study that you're going to be working on. In class on Monday, we began taking some notes on chromosomes and you created a model of the chromosome. We're still going to be working with that tomorrow and Thursday, so hold on to that. But I do want to give you the rest of the notes related to chromosome packing that we started. Now, if you weren't here Monday, you need to get those notes from somebody. And then I have a little case study related to chromosome movement during meiosis to help you better understand both the process of meiosis and how chromosomes are arranged, but also then what ploidy number really does mean and how mistakes to the ploidy number can lead to syndromes and conditions in humans. So if you look at the screen in front of you, you will see that we have our learning objective today is to understand the role of chromosomes and ploidy number in inheritance. So we're going to try to connect those ideas, like how does the chromosome actually affect the inheritance of a particular trait. And the activities we're going to do is we're going to finish our notes on chromosomes and chromosome packaging, and then we're going to do a case study on chromosome movement. We'll be using some of these things here. I'm not sure if you guys have seen those before, but that's an example of a karyotype. So in the textbook yesterday, we were looking at a model for how chromosomes are packaged. And if you look at the diagram I have here on the screen on the right, you see at the top, we have the DNA double helix, which then gets twisted around a series of histone proteins. Histone proteins are the protein that is used to package the DNA. When you get 4 histone proteins together, they form a nucleosome. And as the nucleosomes twist, which is we see in this diagram right here, the second diagram, they form a series of these twisted units, nucleosome units, that condense to create a substance called chromatin. Chromatin is just DNA that has been coiled up in a particular way. But chromatin is important, and it's a word you should understand separate from DNA because chromatin is the form that our DNA actually exists in when it's in our cells and not undergoing division. The chromatin in this state is able to be accessed by this stuff called molecular machinery, which is just another way of describing the different proteins that will bind to DNA to do stuff. So there's molecular machinery associated with transcription, certain proteins that come in and transcribe the DNA into mRNA. There's proteins associated with DNA replication, so making a separate copy of DNA. And again, they need that molecular machinery needs access to the DNA. So those nucleosomes condense to form chromatin, but then within that chromatin, there are these looped domains. Basically, the chromatin is packaged in these series of loops. And if you look at this image over here, they're showing you one of those looped domains. It's this little clear loop right here, and it's a piece of DNA that kind of loops out from the main packaged section of the chromatin. And those loops are going to matter as we start talking about gene regulation, so cells deciding to turn a gene on or off so that the cell can specialize on a certain task or to increase the expression of a certain gene because you've drank some milk. The way that happens is by putting those genes out on those looped domains. And if you look in this third image here, you'd see if a gene was out on the top of this loop, it'd be easy for the molecular machinery to access it, to access the DNA, open the DNA up and read the code. But if the gene had been packaged down in a tighter region of those looped domains, the molecular machinery may just not be able to get there. And that's one of the ways that a cell can turn a gene off or turn a gene on, is by packaging it in a different way, making it more or less available. So you should understand how that might work. Finally, the concept of chromosomes comes at the end. And chromosomes really are just the final packaging of chromatin into a form that can be used to move the DNA around during cell replication, mitosis or meiosis. And that's it. Otherwise, the form of a chromosome, that X-shaped structure that we think about, it's really only present when the cell needs to divide. But nevertheless, we talk about chromosomes because it's a way for us to look at the DNA in individual packaged units. But the kind of weird thing about a chromosome is we always show pictures of them. And here's pictures of two different people's chromosomes. When we show pictures of them, we make it seem like they're all separated from each other. But that's not actually the way it works. In reality, all these chromosomes are connected to each other by a thin piece of DNA because the DNA is continuous. It's not broken up into chunks. So really, the chromosome is just a way for us to understand the different chunks of DNA. On to the case study. The case study you're going to be completing today, after you finish those notes from the page before, is going to look at karyotypes like this. This is a karyotype. It's a squashed cell that was stopped at metaphase 1, and then the DNA of the chromosomes, the packaged chromosomes, were removed from the cell and dyed with a stain so that we could see them. Now, when we look at them, sometimes it looks like, like in this one here, this must be the X shape, right? But that's not really the way it works. Because of the staining technique and just because of the resolution of the camera, each of these bands here, or each of these pieces, is one set of replicated DNA with two sister chromatids joined at a centromere. So we have two of them. That makes sense. There's the chromosome from the mother and the chromosome from the father, and same for each of them. If you get a really good image and you look closely enough, you can start to see the X shape in each of them. So I've zoomed in on some chromosomes here. And if you look closely enough, you can see either side where there actually are two parts, and it forms that X-shaped chromosome that we're used to seeing in drawings. They're just in real life, it's hard to see them. The other thing that's interesting about this is the bands on here, the dark bands. Those are dyed sections of genes that are rich in genes. So it shows us where the most important genes are. And you'll notice in general, any two chromosomes pretty much have bands at the same location. And that's because the mother's copy of the chromosome and the father's copy both carry the same genes in the same location. And so, of course, they would be dyed similarly. Okay, so you're going to open the case study link. And you're going to go to a page that looks something like this. Whoa. Gotta zoom in here. So it's going to be called Case Study, Chromosomal Movement During Meiosis and Impacts on Development. And it's pretty self-explanatory. You're going to do some background research on some key terms and then compare and contrast them. You're going to try to learn about the concept of gene dosage. And then you're going to look at some karyotypes, some sections of DNA, and be asked to figure out a certain, some information. Basically, I'm kind of assessing, do you know how to talk about chromosomes? I can see that I'm going to need to adjust this font here a little bit. Well, I will fix that before you guys look at it. Let's see. Okay, and then the second part is all about meiosis and how those chromosomes are moving during meiosis. And you're going to be asked to identify whether the DNA, the genetic material, is in the form of chromatin or chromosomes. And then furthermore, what's its ploidy number is. And you're going to need to be careful. It's going to talk a little bit about how we name chromosomes. So if you have just one copy of the mother's, genome, we call that a chromosome, right? But if you have replicated that genome and they're still in sister chromatids connected at a centromere, we still call it just one chromosome. So it's kind of this weird naming thing. Even though there's two copies of it, as long as they're attached at the centromere, it's considered one chromosome. But the moment that those sister chromatids separate from each other, they are no longer one chromosome. They are now two chromosomes. And that affects the ploidy number as you read through these. So you're going to be identifying the ploidy number, and be careful. There's kind of a tricky one in there. Most of it's pretty obvious, but there's one or two stages in there that might catch you if you're not paying careful attention. Complete this for tomorrow. Think about how it could lead to trisomy 13. And yeah, that's it for today. All right. Thanks, guys.